Hi, my name is Orazio and I am team leader at Children's Cancer Institute. The focus of my research is on glioblastoma, because glioblastoma is still one of the major killers for children and adults. It is a very aggressive tumor, and unfortunately the survival rate is really low. Survival rate up to 5 years is about 10%. And there are no real cure. Even the immunotherapy nowadays is not working properly. We have seen very low respondents from the patient. So the focus of my research is try to understand better why glioblastoma is so aggressive, but actually also why glioblastoma hides so well from the immune system. And we've found something which was very novel. We found that a metal like copper is very high inside the glioblastoma cells. And apparently this high level of copper helps the tumor to build a sort of wall and protect the tumor from the immune system. So here in my lab we are using some drugs that are already used for other metabolic disease. They remove copper from the system where it is in excess, like in the cancer cells like glioblastoma, and make the cells weaker. So we are using these kind of therapies because they are already there and used for other disease. So this could help us to translate faster our discoveries in the patient. So here what we have, we are in the cell culture room. And what we have here, this one is a cell culture incubator. So what we do have here is cells, which are cancer cells. This one are glioblastoma cells, right? And in these, they can stay alive in this red liquid, which is called cell culture media, because before we want to kill them, we need to keep them alive, because we need to understand a little bit more how they are, how they behave, and what makes them so strong and aggressive. Right? So they are tiny, so we use microscopes to look at the cells. And some years ago, my group and I, we discovered that the level of copper in these cancer cells is about 4 times higher than in the normal brain. And we discovered that this level of copper is important for the growth of the tumor. So there are already some clinical trials using a technique that we published 2 years ago for imaging tumor by using copper-64. So basically they have copper, which is radiolabeled, and they can track the tumor in the brain of the patient. So this clinical trials is running now in Europe, and they cite as well our previous work as one of the discovery which were important to support the clinical trials. What we want to do is use this excess of copper as a target, so we can kill specifically the cancer cells by removing copper from the cancer cells. So we need your help to go ahead with this novel project, because it's very difficult to find as well funding for new ideas. They are risky, and because they are too novel, it's difficult to find someone who want to invest on it. But in reality is that if we don't try new things, nothing will move forward. And now it's been 30 years that basically the rate of mortality for GBM is almost the same, and we cannot go ahead like that anymore. We have nowadays technologies, and we can win together this challenge. So I want to thank you for your support, and I invite you to come and visit our lab and see what we do here daily. Thank you.